This video is to show you how to do visualizing microbes, how to perform a gram stain. You use your gram stain reagents, you use the fixative and decolorizer. You need a slide, and I'm just using the tip box to hold the slide and I don't spill all the dye on my desk. So to start with, I take a pipette tip and scrape the inside of my cheek and then put it on the glass slide. You can make it pretty large, and you want to wait to make sure it's completely dry before you continue. Once it's completely dry, you need to fix it. So you take your decolorizer, which is the same as the fixative, and pipette flood the stain with the fixative / decolorizer. Leave it for about 30 seconds. Then you let the alcohol drop off, ethanol, and use your crystal violet. Again, cover the area with your cells with crystal violet. Move it around if you need, if it's larger. Then leave for 30 to 60 seconds. After the time is over, just tip off the crystal violet and use the iodine to so it can form a complex with the crystal violet in the membrane. Again, make sure it's evenly distributed. Again, leave for 30 to 60 seconds. The next step is to decolorize your sample. That means to get rid of the color of the gram negatives. Gram positive cells will remain blue at this stage. Take your decolorizer, using a pipette, you can flood the smear until no color comes off. We also use the glove pin. Last step is the counter stain, the saffron. Flood the area where your cells are. And leave for 30 to 60 seconds. And then for the last step, you wash it with water. Once all the dye is off, you can blot it with your Bendless paper. Just put it in between and tap it gently. Now you're ready to look under the microscope. What you will see under the 400 with the 400 magnification with your 40x lens is reddish pink stained cheek cells. They are unevenly shaped, and you probably see little dots of bacteria in the background. If you would have a 100x magnification on your microscope, you would see larger cells, different gram-positives and gram-negatives, as you can see here. And also you would see cheek cells associated with your own bacteria, which is perfectly normal. So now you learned how to do a gram stain.